Today I'll talk about chemotherapy. Chemotherapy is a type of cancer treatment that uses specialized medicines to destroy or stop the growth of cancer cells. Chemotherapy can be given to cure cancer, to control cancer growth, or 3 for palliative care, that means to reduce the pain or symptoms. Chemotherapy can be given before surgery, which is called neoadjuvant chemotherapy. It can also be given after surgery, which is called adjuvant chemotherapy, or also used as the main treatment in advanced disease. It is given in cycles, treatment followed by rest, to allow normal cells to recover between these rounds. So how does chemotherapy work? Chemotherapy destroys the cancer cells by intercepting the cell cycle. So the cell cycle is the biological process that cells use to grow, copy their DNA and then divide. Because cancer cells multiply at such a fast, rapid rate, they are highly sensitive to such an intervention. So there are many ways in which chemotherapy can work. One, direct DNA damage. It chemically binds and breaks the cancer cell's DNA strands, making the genetic blueprint unreadable and uncopyable. 2. It can mimic fake nutrients, so it will act like a building block and the cell will absorb it by mistake, and this leads to jamming the DNA replication process permanently. 3. It can lock the DNA zipper, so it blocks an enzyme called topoisomerase, which is required to unwind the DNA helix. This leaves the genetic material tangled and 4. It can freeze cell division. So it stops the cell from multiplying by breaking the internal framework of the cell. 5. Shredding with free radicals. So chemotherapy can create highly destructive, unstable oxygen molecules that physically tear apart the cell's outer membranes and internal structures. 6. Inserting DNA obstacles. It physically wedges itself directly between the rungs of the DNA ladder, which changes its shape so the cell cannot read its own instructions. 7. Starving vital proteins. It destroys specific amino acids that are used to make proteins circulating in the blood, starving the cancer cells of nutrients that they cannot make on their own. And 8. Triggering apoptosis, which is natural cell death. So as a backup, the severe stress caused by any of the methods can force a cell to activate the built-in self-destruction sequence. The dose and unit of chemotherapy. So chemotherapy dosage is the specific amount of anti-cancer medicine that is given to a patient, and the unit is milligrams, the most common unit. Milligrams per square meter measures how much drug is needed based on your body size. So how is the dosage of chemotherapy decided? 1. Your body size. Most often calculated using your height and your weight to ensure you get enough to fight the cancer without causing severe harm. 2. The type of cancer. Different cancers require different drugs and different strength of drugs. 3. Patient health and blood work. Doctors check your liver, kidneys, blood cell counts before every treatment. If your body needs time to recover, the dose may be lowered or delayed. And just like radiotherapy, even the dosage of chemotherapy is not given continuously, it's given in. Today I will talk about a very interesting phenomenon in chemotherapy called collateral sensitivity. So what is collateral sensitivity? When a cancer cell mutates to become resistant to chemotherapy drug A, let's say, accidentally it becomes sensitive to another chemotherapy drug B. This is not coincidental. This happens through 3 major biological mechanisms. The first one is energy exhaustion. So here, to survive chemotherapy, cancer cells evolve efflux pumps. These pumps act like tiny bouncers. They grab and throw the chemotherapy drug out. But to throw the drug out, a lot of energy is required. A lot of cellular ATP is required. So the cell burns energy so furiously that it creates toxic products inside itself, like reactive oxygen species. So now doctors can introduce a second drug that stops the cell from neutralizing these toxic waste products. An analogy for this would be, there's a boat, the boat has a leak, so water is coming in. So people install a very expensive pump that can pump the water out of the boat. But this pump uses a lot of energy and generates fumes inside the boat itself. So now even a small match would light up the entire boat. The second mechanism is structural changes. So here a cancer cell would change its shape so that a drug cannot bind to it. So for example, to resist a drug like paclitaxel, the cancer cell mutates its structural proteins beta tubulin. So now paclitaxel is blocked, but the shape of the cell membrane is changed in such a way that another drug called verapamil can now bind to it and kill the cell. The third is metabolic rewiring. So here, to survive an attack on its normal digestive or metabolic pathways, the cancer cell completely reroutes how it processes nutrients and energy. So the cell will shut down the usual chemical pathway to avoid processing a chemotherapy drug. And it opens up an alternative pathway to process nutrients. Now this alternative pathway is very fragile and inefficient. So if a secondary drug is used which targets this alternative pathway, the cancer has nowhere left to turn and it starves. A simple analogy here would be, if during a war, a main road is blocked so that the enemy cannot come in, but that main road was also the source of food, nutrients, water. So instead of that, an alternate hill trek is opened up for food and water. Now the enemy just has to use a boulder or something to block that alternate route and everyone is stuck without any food or water. That's what happens to the cancer cell. Today I'll talk about a very interesting phenomenon in chemotherapy called synthetic lethality. So this basically means that there are two genes required for a cell to function in a healthy manner. That is a normal cell. But if one of these genes is stopped due to some reason, the cell still has a backup gene 2. But if both these genes are stopped or mutated in some way, then the cell dies. So there are three main mechanisms how synthetic lethality works. The first one is, one of the genes is mutated. That's what happens when a normal cell becomes a cancer cell. So one of the genes is already mutated, and the second one is inhibited through a chemotherapy drug. So that means gene 1 is not normal and gene 2 is inhibited through a drug. So both genes are not there and the cell dies. In the second method, both the genes are mutated, double mutation. So the cell once again does not survive. The third mechanism is when one gene is overexpressed and the second gene is inhibited through a chemotherapy drug. So once again, the cell loses 2 copies of a gene and cannot survive. I'll explain the first mechanism through an example. The first mechanism is mutation plus inhibition. So in breast cancers and ovarian cancers, when a normal cell becomes a breast cancer or an ovarian cancer cell, it develops a mutation called BRCA. BRCA is responsible for double-stranded DNA repair through homologous recombination. Now the cell has lost its ability to repair double-stranded DNA breaks, the breast cancer or ovarian cancer cell, and this is the first hit. But the cancer cell does have a backup repair mechanism through PARP, P-A-R-P. So now there's a chemotherapy drug which is a PARP inhibitor. So this drug paralyzes the PARP repair crew, which normally can fix single-stranded DNA breaks. Now because PARP is paralyzed, it does not work, so single-stranded DNA repair does not happen and the cell tries to copy DNA to divide. These minor scratches turn into massive double-stranded breaks, and this is hit 2, and hit 1 plus hit 2 is what causes the cell to die. Today I'll talk about the types of chemotherapy. So there are 5 major types. The first one is alkylating agents. So here what happens is an alkyl group, which is CnH2n, that means carbon a certain number and hydrogen 2n plus 1 of that number, goes and attaches to the guanine base of DNA. DNA has 4 bases, A, T, G, C. An alkyl group will go and attach to the G base. And this does 2 things. One, it will form DNA adducts. That means a piece of DNA that is bonded to a cancer drug. And 2, it will form crosslinks. That means that the 2 strands of DNA get glued together. And this prevents DNA strand separation. And for replication to occur, DNA strands are supposed to separate. So therefore, DNA cannot replicate. The second mechanism is antimetabolites. So these are substances that mimic and act identical to the raw building blocks that a cancer cell needs to grow. So they're not actually the building blocks, they're acting identical and mimicking those building blocks. And now the cancer cell absorbs them, thinking that these are important, and they end up jamming the reproductive machinery and starve the cell to death. The third mechanism is anti-tumor antibiotics. So what these do is they go inside the DNA ladder and generate damaging free radicals, highly destructive oxygen molecules, and these molecules induce double-stranded DNA breaks. That means snapping of both the strands of DNA. The fourth mechanism is topoisomerase inhibitors. So topoisomerase is a very important enzyme for cutting, untangling and re-gluing DNA during copying. And topoisomerase inhibitors will bind and trap topoisomerase. So therefore, religation, that means gluing the cut DNA strands back together, cannot occur. And the fifth mechanism is mitotic inhibitors. So mitotic inhibitors go and bind to tubulin protein, which is an important building block to help the cell's internal skeleton basically. And therefore, the building up or breaking down of cellular scaffolding that pulls a dividing cell apart cannot occur, and this freezes the cell in a particular phase of the cell cycle called mitotic metaphase.